Thank you so much for joining us today. Before we kick this off, I do want to point out to the audience that we welcome questions. Please use the Q&A section at the bottom of your screen. Type in questions as they come to you, and we will address those after today's presentation. So it's my great pleasure to introduce Pankaj Khanna, CEO, Novel Bio. Pankaj, the floor is yours.
Thank you, everyone, for your time today. So a little bit about myself. I have about 2 decades of experience in the biotech space. Most of the experience has been in technology development and commercialization. And Novel Bio is my third venture in this space. The other two ventures are focused on manufacturing setup for different biotechnology companies and also regulatory compliance consulting, where we provide consulting to biotech companies how to navigate GMP manufacturing and FDA compliance. So I'm going to share my screen now. Wonderful. So we call it a better way to DNA, the next chapter in microbial production of DNA. So the idea of Novel Bio came when I was working during the COVID pandemic and actually setting up manufacturing facilities for plasmid DNA for other biotech manufacturing firms, and realized that at that time, plasmid DNA's lead times were going into years, and people were using 50 years old technology, and the industry was ripe for innovation. So from there, 4 years ago, we started this company. So let's start with what is DNA? So DNA is a software of life. Everything that we do in biotechnology starts with the DNA process. So whether it is any therapeutic medicine, it is mRNA vaccines, direct DNA therapy, cell and gene therapy, or industrial biotechnology products like cosmetics or detergents or enzymes, everything starts with DNA. And the process of making DNA is called molecular cloning. And molecular cloning involves a bacteria called E. coli. So that is the heart of the manufacturing process. And E. coli acts as a small factory that kind of makes DNA in small quantities. And this is the technology that has been used for the last 50 years. Since the 1970s, people have been using E. coli to make DNA, not because it is the best bacteria for this process, but because it was the only available bacteria that people knew how to work with in the 70s, and they started making DNA from it. And since that time, they just have been using it just out of convenience. And from that, the last 50 years, the industry has really evolved, the biotech industry. The DNA designs have become very complex and challenging to make, but people have still been stuck with the 50-year-old technology. Because of that, there are a lot of pain points in the industry now. So the major pain points are that the E. coli is very slow to grow. It takes a lot of time for E. coli to make DNA, and that was one of the reasons why the lead times during the pandemic, because mRNA vaccines were using a lot of DNA, was going into years. The yields are low, so it takes a lot of bigger batches to make small quantities of DNA. And also the failure rates are high because the designs are complex, and E. coli kind of struggles with that. So because of all these reasons, the cost of DNA is very high. A gram of GMP grade DNA will sell for about $1 million. And as it is the starting material for these cell and gene therapies, this makes the therapies very expensive. And that is not only a problem from the patient's point of view, because they cannot access these life-saving medicines. This is also a bad business case because it takes a lot of effort and money to do the drug development of these medicines. And then if you cannot provide it to the clients because or the patients because they cannot afford it, it kind of defeats the business case itself. So industry is really looking for a solution to lower the cost of DNA and improve the timelines. So there is a lot of demand for this kind of solution. So if you look at the size of the market, there are two distinct markets. One is that where people sell vials of or small tubes of E. coli to make the DNA. And that is used for mostly R&D purposes, where the scientists do not know which DNA is going to work for their application or which DNA is good for a certain therapy. So they make a large number of designs and buy lots of tubes of E. coli and then do the molecular cloning to make DNA. So that market itself is about $2 to $2.6 billion. And all these companies, Thermo Fisher, New England Biolabs, or Merck, all they are doing is selling small tubes of E. coli to the users. And the industry is growing. It will be around $3.6 to $4.4 billion by 2030 to 2032. The other sector of the industry is where we do large scale manufacturing of DNA. So once scientists find out that one particular design has a use case or better design to take forward, they want larger quantities of that DNA. And they want various quality grades of that. So they start with R&D, then they go with high quality for phase 1 clinical trials, and then GMP for phase 2 and commercial manufacturing, phase 2, phase 3, and commercial manufacturing. So that market is a separate market. We call it CDMO operations, and that market is rapidly growing. It's about $2.1 billion right now in 2024, and it will be around $6 to $10 billion in a couple of years. And the major players in that is Aldebaran and Thermo Fisher. And again, both of these markets are separate markets, but they are joined by the common thread of E. coli. There is no other technology. Everybody in the cloning space use E. coli. Everybody in the large scale manufacturing of DNA also use E. coli. And that is where we come in. So our basic premise was that everybody has been using E. coli for 50 years. There has been no technology differentiation, a lot of pain points. And over the last 50 years, we have actually found a lot of new bacteria, which are fundamentally superior as compared to E. coli. So why don't we change the host itself and look for a bacteria that can do a better job at making DNA? And we had some parameters to kind of choose the right bacteria. The first parameter was that it should be really fast growing because the timelines matter a lot in this. Second is that it should be high yielding and it should provide high quality of DNA. The quality should be better than what we get with E. coli. And the last was that it should be drop-in compatible, that nobody should be, should have to build a new facility. It should work with the existing equipment, with existing DNA designs. So nothing new, just use our platform and our bacteria and you're good to go. So those were our criteria, kind of that we chose. So we call it the NBx platform that we have developed. And when you look at the speed, our bacteria is the fastest growing bacteria on the planet. There is no other bacteria that grows faster than our bacteria. And typically a cloning process takes about 3 days with E. coli. With our bacteria, you can finish that in about 15 hours. So when you're testing hundreds of thousands of designs or thousands of designs, it really accelerates your R&D process. And we have already launched a product on that. We're getting excellent feedback that it's really helping the researchers to move their R&D faster. So that's a great advantage with our platform. The other is that the yields are also pretty high as compared to E. coli. So here we are comparing for a standard DNA design, our platform versus 2 of the leading competitors of E. coli strains. And you can see here that our yields are much better as compared to E. coli. And the quality is also better. So E. coli struggles, as I mentioned before, with complex DNA designs. It makes impurities, unwanted impurities. And this image kind of shows that, where our bacteria or our platform is able to make very clean product, while one of the strains of E. coli struggles, with the quality as well. So in summary, we have a platform that is faster, higher yielding, and better quality as compared to E. coli. And it is also drop-in compatible. So we have tested the platform against various DNA designs. We have used it in the same commercially available equipment, and it works beautifully there. There have been no challenges that we found. And it works with all the standard available equipment upstream and downstream. So all fermenters and downstream equipment are the same that are used for commercial manufacturing of E. coli. We can use with our platform as well. So once we had the platform developed, we actually am a great fan of testing the platform with the clients with real world examples. So I have a deep network within the biotech industry, and I reached out to some of my friends and I said, hey, if you have any DNA designs that you have problems with E. coli with, send us our way. We want to test it against our platform. So people started sending hundreds of DNA designs to us. And we started this process about 2.5 years back. And this is a small data representation of the entire spectrum of designs that we have tested against E. coli. And this actually graph represents different types of DNAs of different applications. So some are for cancer drugs, some are for mRNA vaccines, some are for synthetic biology, making, enzymes, some are for viral vectors. So if we have kind of tested the whole spectrum of the design, space for DNA, and you can see here on an average, our yields are always higher as compared to E. coli. And one thing we found very interesting that we hadn't realized before, that as we are using a different bacteria, we can make the DNA that is toxic to E. coli or E. coli cannot produce. And that's a big deal because before our platform existed, if researchers had a very good hope on a certain DNA design and it was bad for E. coli and E. coli could not produce, they had no other option but to actually mothball or actually change the design itself, because there is no other technology out there that can make the DNA itself. So now they have another option. So if they like certain DNA design and it doesn't work with E. coli, now they can work with us. And that opens the way for new therapies that could not come to the market before to come to the market now. And we have existing examples I'll demonstrate in the subsequent slides, where people have been able to take their designs forward using our technology already. So the last piece for us was we kind of tested a lot of DNA designs that worked beautifully in our platform, but is it scalable to commercial level or not? And we put the bacteria in a standard bioreactor, 5-liter bioreactor, that is our commercial scale. And we found that right off the back, it gave us about 2.5 times the yield as compared to E. coli. And we are optimizing the process right now, and we're expecting to get 8 to 10 times the yield, similar to what we get at a smaller scale as we optimize the process. I've been in the process of or in the scale of business for the last 20 years, so I'm highly confident that we'll be able to achieve 8 to 10 times yield very quickly. We're already, this is the older data. We're already about 5 times already, so we are gearing towards 8 to 10 times. And this is using the standard bioreactor. And we also did a downstream purification work with Sartorius, which kind of demonstrated that with standard purification system of using from Sartorius, which is the industry leader in this space, we can produce a material of right quality, which is acceptable to FDA using Sartorius equipment. So it is completely scalable from upstream and downstream using commercially available equipment. So how does that speed and the yield and better quality lends itself into a business case? Because that's the ultimate test, right? So if you compare our platform and E. coli to make the same quantity of material, it will take about 100-liter bioreactor to make a specific quantity of material. That essentially means that you need about 10,000 square feet of GMP facility, about 20 million in capex, and a lot of OpEx expenses to maintain that facility, because these are clean rooms that needs to be operated continuously, needs to be maintained, a lot of water, high purity water that needs to be generated and maintained. So it all lends itself into the cost of the batch, and that's why it's $1 million for a gram of material. In our case, we can do the same in 5 liters in 1,000 square feet facility. So our capex are way lower. And because of that, our cost structure that we have set up is less than 50% of the cost of what the competitors are offering. And we've already launched our high quality. I will explain more in the subsequent slides. And we're getting very strong response from the industry. So this is solving a key problem and key pain point of the industry of high cost of DNA and completely disrupting a multi-billion-dollar market. So once we had the platform, we started reaching out to clients, and this is a case study for partner success. One of the top 5 animal health company reached out to us, and they said, we are making RNA vaccines for animals, and our mandate is to be able to quickly make the vaccines and as cheaply as possible. If there's an outbreak, we want to quickly react to it. DNA is our bottleneck, as it is always the case with these medicines. So they gave us 5 designs to test, and they said 4 of these designs we can make in small quantities in E. coli. The 5th design is a great candidate for vaccine, but we cannot make it. Can you please test it in your system? And if you can make 3 times the yield, we will be able to license the technology from you for global manufacturing of these RNA vaccines. So we imported all the 5 designs, we put it in our system. We got 10 times the yield on every single design. And we also were able to produce the DNA that they could not produce before. So remember what I said before, where it opens the doors for making new drugs. And this is a clear case there where a client, top 5 animal health companies, able to take a vaccine candidate forward, which they could not before. So we were able to go there in their lab, in their manufacturing setup, and demonstrate the same results in their hands too. So now we have signed a term sheet with them where it's a royalty plus deal, where they will pay us royalty on sale of their vaccines, and they'll be using our platform to make RNA vaccines globally. We are working on a definitive agreement. It is expected to be signed in the next month or two. So as we have developed the technology now, we've started to commercialize it already. We are making DNA with this technology for the last 2 years already in our lab. And I'm a big fan of basically multiple revenue streams, which are kind of complementing each other. So as I mentioned before, there are two different markets. One is people sell small vials of E. coli to make DNA, and the second is CDMO operations. So we are playing in both the markets. And our main market is the CDMO market. That's where our kind of, I would say, bread and butter is. But where we are selling already, we call it the product Cyclone, to make the clone, to do the cloning of DNA. And the intention there is that people can test the technology in their own hands, that generate confidence. It's a new technology. It's a low barrier to entry. We sell a box of those cells for about $400. People can buy. It's very large quantities, but the unit cost is less. And also it acts as a funnel for our CDMO business, because we are giving them only research use, only limited license. So when these DNA that they're making or cloning go into commercial manufacturing, they have to come to us because they used our technology to make the DNA in first place. And we've launched that product already, and we got pretty strong response from the industry already. And then the third piece, the second is the CDMO operation, where we are basically already making for the last 2 years R&D grade plasmid. We started making high quality plasmid from this month, and we will be making GMP grade plasmid from next year. That's our plan. And then the last revenue stream is licensing the platform to strategic clients, just like I said, the top 5 animal health company. So similar to that, if somebody else comes to us that is a drug producer and they want to use our technology to make DNA in their own lab, we are kind of open to that as well. So now coming to the IP. So this is pretty solid IP protected technology. We have filed 3 patents already on it. And we have great first movers advantage. We know this bacteria more than anyone else, and not much has been published about this bacteria so far. And beyond the patents, we have a very deep trade secret knowledge as well in this space. So anybody who wants to enter this space has about 8 to 10 years of work in front of them, and they still cannot come close to us because we have filed foundational patents. The wild bacteria really cannot make DNA. We have engineered the bacteria and mutated the DNA of the bacteria for it to be able to make DNA. And those foundational patents prevents anybody else from entering the field using this bacteria. So we are already making revenue. We are a revenue making company. This platform has been commercially deployed to great success. As I mentioned before, we have been making revenue for the last 2.5 years. And first year was just to try out the platform. We had no business development person on board. I just reached out within my network, and we were able to generate about 100,000 in revenue in the first year itself. The next year, we hired one BD person who was wearing multiple hats. We had to generate a lot of collateral, marketing, and all of that. And we were able to double our revenue, more than double it. And now this year, we have another BD person joining for the last 4 or 5 weeks, and we are seeing very strong growth. We expect to finish this year with about 750,000 in revenue. And that is just with very limited business development resources and very limited lab space, which is also doing a lot of R&D work as well. So we launched our Cyclone cloning product in May this year, have already shipped to about 30 plus clients, and the customer feedback has been amazing. One of the researchers in cancer research at NIH came back to us after using our product and said they were trying to clone one DNA for cancer treatment drug for the last 6 months with E. coli, and they could not do it. With our product, they were able to clone it within 2 days. So that's another success story. So we're getting these success stories regularly now. Not only that we are cheaper and better, but also we can make DNA and bring new drugs to market that were not possible before. At the same time, we just launched our high quality this month, and our cost, and this kind of lends itself into how this is a great advantage to the patients, great business case, but also great advantage to the patient. So we got our first purchase order within 2 weeks of launching high quality without any business development work. And the reason was a client came to us. It's a compassionate ground case study, phase 1 case study, and it's a terminally ill patient looking for this therapy. and the plasmid DNA cost was preventing them to move the plan forward. And our quote was less than 50% of what the competitors were offering. We got the PO within a week. And now we are making that DNA that will allow this therapy to move forward. So we're getting great response from the market. So this technology, in summary, doesn't exist on a PowerPoint slide. This technology is real, has been deployed, making revenue with repeat clients, with customer feedback on multiple points. So this has been de-risked to a great extent now. And what we are looking for is to grow to the next phase. Another feedback we got in the case study was that a client of ours, which is a repeat client now, it's a large publicly traded company with 4 billion market cap. They were using the industry gold standard vendor that holds 40% of the DNA market. And they gave the same DNA design to make to them and to us. And we were not aware of that. That's a bake off. But we just did our work. And within 3 weeks, we delivered the DNA to them. And they were going to make viral vectors with that. They were able to successfully make it. The other gold standard vendor took 3 months to deliver the same DNA, and it failed the quality testing at the client site. Since that time, they have moved all their R&D grade DNA business to us for the last 1.5 years. They are our largest clients, and they keep coming back to us. We have been gaining one client after another from these gold standard big vendors because our technology is much better, our cost structure is much better already. So these are our revenue projections. 2025, as I mentioned, we should be around 700 to 750,000 revenue. We have very strong growth. We expect to break even next year. We are right now prevented by our lab size. We got about 200,000 in POs in the last 2 months alone, and I had to slow down the business development work just because we are overwhelmed. We need a bigger space and hire more people to quickly ramp up the revenue and a kind of our growth. So that's why this fund raise is for. But we expect to breach about above 40 million in revenue by 2030, and it's not a very capital intensive growth cycle because as I mentioned, our capex requirements are relatively small as compared to other traditional E. coli based technology. So we expect about 15 to 20 million in capital by 2030 to reach this revenue potential. And we are expecting a Series A in the next, say, 12 to 18 months. And the expectation is that we'll set up a GMP manufacturing facility, or expand the facility. And we will have enough clients already asking for GMP material. So we set up the facility and the revenue starts to flow in right away rather than we build a big GMP facility and now we are waiting for clients. So it's a very finely calibrated CapEx expense plan that gives returns very quickly. And the M&A in this space has been pretty active with a revenue multiple of 10 to 30x in the last 5 years. And those M&A activities had no technology differentiation. They were all using E. coli just based on the growth of the market and their size of the revenue. They got that 10 to 30x revenue. We have a great technology differentiation already on that. So these are some of the M&As that happened. So Danaher acquired Aldevron, which holds about 40-50% of the market for about $10 billion in 2021. Charles Rivers acquired Cognate Bioservices for 875 million. Ajinomoto is a Japanese player. They acquired Forge in US for 620 million. Oriciro was acquired by Moderna at 85 million. And Express Bio is a undisclosed, but again, from the grapevine, we know it between 10 to 30x revenue multiple. So there are multiple, and all these are in the last 4 or 5 years. So what are the major risks for us? The major risk for us is that we have a new bacteria. The adaptation or adoption of this new technology might take some time. And I apologize for this. I don't know, somehow my this format has been screwed up. So some fonts are there, but I can send that presentation to you guys if later on if you require it. Apologies for this mess up. But coming back to the risks, one is the slow adoption of this technology. So I think that risk is greatly behind us to a great extent because we've already launched the Cyclone product for cloning, and we're getting a great uptake from the market already. And we already have deployed the platform at R&D grade scale. Clients really do not care. They care about quality, cost, and timeline. So as long as we are meeting that or exceeding that, they're happy. So we don't see that issue. For high quality and GMP, we have a clear regulatory strategy on how to approach that. Again, this is a starting material. This is not a finished drug product. So we don't have to deal with FDA at all, actually. We have to deal with our clients. So from that perspective, the FDA risk is pretty low what we are doing. But I come from an FDA background, so I have a pretty good understanding of we have a plan in place how to tackle that as well. The second is technical challenges. So the platform is great with about 90-95% of the designs. It does struggle with some specific design cases where E. coli still does better than us. So we are doing, and that's a small portion of the market, and we are doing the optimization and evolving the technology to kind of overcome that. But for our CDMO operation, we have both the technologies available. We have a great E. coli technology as well with us. So if any client comes to us and we have that fringe case design where our platform doesn't work, we can always cater them with E. coli as everybody else is catering to. So we don't have that disadvantage. We actually have two tools in a toolbox rather than like everyone else who have only one tool. And the last piece is optimization of the scale-up process. So we've already demonstrated that it is 5 liters. It works at 5 liter, which is our commercial process. We might migrate to 10 liters for large GMP manufacturing, which is a very low risk scale up from 5 to 10. At the same time, we've already demonstrated our downstream work is completely scalable as well. And our process optimization is already ongoing. And I think that risk is also pretty well taken care of. So we are raising the seed round for $5 million. And as I mentioned before, most of this money is actually going towards revenue generation. The technology has been developed, commercially deployed with great results. So a lot of what we're looking for is expand the lab space, hire more people for manufacturing, and that will directly contribute towards revenue. And one thing to mention is we are at a great inflection point as we are about to sign the licensing deal, as I mentioned before. Plus, we have launched the high quality. Our revenues should expand very quickly. So from that angle, this is a great investment at this point in time. So I think it all boils down to the team that we have. So as I mentioned myself, I have about 20 years of experience in the GMP manufacturing space, built a lot of facilities for others, operated CDMO facilities as well, understand this space really, really well. Matthew Weinstock is our CTO. He has about 20 years of experience in molecular biology. He trained under Dan Gibson, was also CTO of AppSci, where he joined early and took the company public. So pretty senior executive and researcher in this field. We have Melissa Patterson, who is VP of Business Development. She has multiple experiences in multiple large organizations in alliance management and business development. And Konstantin Rodionov is our COO. He has experience in multiple startups that spans across real estate and technology development and all that. And we have a very strong advisory board as well, which include ex-deputy director of US FDA, Jeff Baker. We have Anthony Hitchcock, who is a veteran of plasmid DNA manufacturing, used to work with Charles Rivers Laboratories before. We have Beth Thompson, who used to be the COO of Aldevron before it was sold to Danaher, and Todd Peterson, who is a very accomplished researcher in the DNA space as well. So thinking about the future, again, I apologize for this. I don't know what happened here. I think when I put it in the presentation mode, it kind of screwed up the fonts here. But so plasmid DNA is our starting point for this technology, and that is where our focus is. But we also know that E. coli just does not only make DNA, it makes many other things apart from DNA. It makes industrial enzymes and chemicals and many other things. Our bacteria is fundamentally superior as compared to E. coli, and we have the means and methods to develop those other applications. And one of the examples is that where we're not even looking, we launched our Cyclone product in a conference in May, and one of the senior vice presidents of a top 100 consumer health company reached out to us, Fortune 100, reached out to me and said they've been trying to use this bacteria for the last 2 years to valorize their waste stream. And they could not do it because of some technical challenges, which we had overcome to launch our product. And we are not looking in that direction, but it was kind of a partnership on the table with a great company. So we basically, they approached us, and we are working with them right now to do a trial project with them to demonstrate how our bacteria can help them in what they are trying to do. So these are the applications and different things that we're not looking for right now, but they will come in the future as the company expands. Same goes for protein expression. We have a protein expression strain in development. This bug can actually do a great job at protein expression. Same, faster, better, cheaper, and it has been demonstrated before. So from that perspective, there are multiple verticals in the future that can come from this base technology. So why we are a great investment and or a great company to work with? It is because we have a technology differentiation. We have a great team. We have demonstrated that the technology works. We have traction in the market with real customers, and our IP is rock solid. Very hard for anybody to kind of break into this field with this bacteria because we have a potent mix of patents and trade secrets which are not available anywhere. So that's the end of my presentation. Thank you so much for your time.
I'm going to ask you to stop sharing your screen, and we will go into the questions. And some of these you may have touched on, so sorry for any repetition. So it sounds like there is a really large market for this. Do you have current clients using this technology?
Yes. So we have about 10 clients already, and that is using our CDMO operations. Then we have shipped our Cyclone product to about 29 clients already. So we have a large customer base and growing every day.
And can you just expand on what are your primary customer segments aside from the CDMOs? Research labs, gene therapy firms, and how is this evolving?
Yeah, so the most of our clients are from the therapeutic world of cell and gene therapy, mRNA vaccines, as well as direct DNA therapies. That's our core of our client base. Then there are other companies for our Cyclone product that make DNA at large quantities. So that do cloning in large scale, like Twist or IDT. They are our clients for our product of Cyclone. And then there are smaller clients around academia. They're also using our services. And the final are the synthetic biology firms who are making, enzymes or other chemicals. So, but the core of the business is the therapeutic companies.
Thank you. And you mentioned a significant growth rate of the DNA manufacturing market. What applications do you see growing fastest, and how will you capture those customers?
That's a great question. So, cell and gene therapy and mRNA and direct and DNA therapy is the future. There's no doubt about that. That is I call it the third revolution. First was the solid oral dosage where we started making pills. Second was mammalian cell culture to kind of uh, some treatments or limited treatment options for cancer. Now we are actually in the third revolution of biotech where we are cell and gene and mRNA and direct DNA. Now, within these three different, I would say, methods or technologies, they will see their own challenges and ups and downs. Viral vector technology might see one day some successes, some failures. mRNA, same way, direct DNA, some way. But the good news for us is we sit at the bottom of that value stream. So for all of these, they need DNA. So the demand might shift from viral vectors to mRNA for some time. Well, that's okay for us because they still need DNA. And the same goes for demand might shift from mRNA or direct DNA therapy to viral vectors. They still need DNA. So in summary, to answer your question, we expect strong growth in the therapeutic segment, and we expect there are more than, I think, 2,000 clinical trials happening in this space right now. And in the cell and gene and mRNA space, we expect rapid growth within this broad segment of modern medicine. And there'll be some adjustments within the subcategories of these modern therapies, but we are not impacted by it because all of these subcategories, the whole segment, requires DNA as a starting.
And as you transition to GMP grade plasmid production, what specific technical or regulatory milestones need to be met? And when do you think you would hit those?
Yeah, so that's a great question. So for GMP, just to give you a very quick high level overview, what is the difference between R&D to high quality to GMP. So R&D is when we make DNA without any documentation, and we just make it. It should pass the test and we're good to go. Nobody asks us, how did you make it? There's no documentation proof for it. Then the second is high quality, where we have to document everything. Which material did we use? How did we make it? We call it batch records. Operators need to be trained. We call it, the full spectrum is called quality management system. And then the last piece is GMP, which is one step above, where we, instead of making the material in a regular lab, you do it in a clean room where the environment is also controlled now with HEPA filters and all that. So those are the three different quality grades. We have already been doing the R&D grade plasmid. We started doing the high quality DNA already. So we have set up the quality management system, batch records, SOPs, and all that is all set up already. I come from that world, so we know how to kind of skin that cat really well. And then the GMP will involve setting up a GMP manufacturing facility, which is like the clean rooms, and qualifying them. So it's a small step above where we are right now, but it requires, from the technical perspective, but it requires capital. So you need to build clean rooms, which are expensive to build. The good news is our capital requirements are about 1/10 of our competitors, but still there are capital requirements. So that's how we're going to kind of progress across the quality grade. And we are using a new technology, but the good news there is that because we are a starting material, we are not a drug product. People make drugs from our material. We are not a drug itself. So we do not need specific approvals from FDA to make DNA. So that's the kind of makes it easy for us to navigate this.
Thank you. And so kind of following that, can you talk about your IP strategy and what aspects of your technology are patented or defensible, and talk about your competitors in the space?
Yes, yeah. So our technology, the IP, our IP strategy is very clear. We have only patented genetic mutations in the wild form, which kind of blocks the field from others from entering. And also those patents we can easily enforce because we can, sequence the genome and find out if they've used the same mutations as us. So it's very easy to enforce. Our trade secret strategy is driven by everything else that is not enforceable, like our media recipes. How do we, different types of solutions we treat the bacteria with, which anybody can use and we will never find out. That has never been that has not been put into patents, and those are all trade secrets. So between the two, it's a very potent combination where people cannot just come and read our patents and start copying. They just cannot, because there are a lot of things that are not in the patent itself. So that's the overall picture. Now, coming to the competition, there is no company that is using any other bacteria as compared to E. coli. There are companies like Touchlight and 4-Base Bio and some others who are using synthetic, like cell-free process to make DNA, where they're using enzymes made by the bacteria, mostly E. coli, to make DNA. So it has some small application and advantages over a bacterial-based process, but also disadvantages. So the advantage is that you don't need to use bacteria, which sometimes is a problem using in a GMP facility, and you can do it in a very small footprint as compared to, say, E. coli. But that's where the advantages stop. The disadvantages of that technology is, first of all, it still uses bacteria in the first step. You're not getting away from bacteria. So if for the first step of DNA making, you still use E. coli. So if E. coli cannot make it, you cannot take it forward. The other aspect is you're using a lot of enzymes which are made from bacteria. So it makes, and enzymes are way expensive than sugar and water that we use in a bacterial process. So sugar and water is the cheapest thing. So the cost is much higher. So you already have a problem of cost of DNA, and you make it worse by using enzymatic process. Now, on top of it, when you want to scale, it's very difficult to scale because it's very hard to use so many enzymes in a very precise way at a larger scale. So they get quality problems at scale. So those are the three main downsides of synthetic or cell-free platforms, and they are the only viable client or competition we have. And we've already seen it where the cell-free guys are quoting against us, and we are winning every time because their cost is even higher than E. coli platforms. So it really, they're not breaking the market. They're not disrupting the market from where it needs to be. The real disruption is in cost and lead time, and their technology does the opposite, you know.
Okay, thank you. Do you have an exit strategy, strategic acquisition, IPO, or continuing as a revenue generating private company?
So yeah, we have an exit strategy. We are already looking at potential acquisition by a large player, and we have some feelers already from them. They want to look at our technology, but we are cautious at this time because we want to basically have some more successes to extract the maximum value. So I can only disclose this much, but we have interest from large players, and we're open to acquisition as well. But my viewpoint is that we need to have bigger success and market to cause some pain to the potential acquirers and demonstrate the value of our technology to extract the maximum value out of this enterprise for us and for the shareholders.
And what are your anticipated margins for the different revenue streams?
That's a great question. So our margins on and they're different at different different services. So our Cyclone product has a gross margin of around 80%, pretty high gross margin, and we are keeping the cost similar to E. coli. We are not charging a premium, although we have a premium product. Then our gross margin R&D is about 10 to 15%. And the reason for that, sometimes we even get 25%. It depends on the client. And the reason is that the R&D grade market is dominated by Chinese players, and it's very hard to compete on cost with them. Most of the American companies are unable to compete. We are able to compete with them and still extract some margins out of our work because of our technology advantage, our high productivity. But when we go to high quality and GMP, the Chinese players disappear because at that time, people get very, very serious about IP because now they're getting into clinic and they don't want that IP to be shared by outside the country. So from that perspective, as soon as that kind of element disappears, we are around 50% of the cost of the competition, still maintaining more than 50% gross margin on high quality and GMP.
Great, thank you. And what is your break-even point? What is the projected margin on the 700K of revenue? And then we'll get into your investment ask.
Yeah, so we expect we have been kind of we'll almost triple our revenue this year. We expect similar trajectory or slightly higher because high quality is a higher grade material and POs are very large. Size is large. R&D grade POs are like $5,000, $10,000, $20,000. Our high quality POs are $100,000, $150,000 on each PO. So from that angle, we expect to be around between 2.5 to 3 million in revenue next year. And that will be our break-even point with keeping the operations lean. And from that point on, the capital will be used for growth in a responsible way. So that's our overall picture for this year and next year.
Great. And the 5 million seed round is at what valuation? And is it straight equity or convertible debt or other? And what will be the use of your proceeds?
Yeah, so it's a standard Y Combinator safe at 20 million pre-money. We already have commitments, so everybody is getting the same terms. Some safes have already been signed. So we are 20 million pre-money with zero discount, and it's a standard safe. So that's the first question I have. If you can please the second question that you asked, I forgot that. Missed that one.
Use of proceeds.
Yeah, use of proceeds. So we are right now doing all of this out of a 1,500 square feet lab. We're doing all the R&D. I have like 9 people, and we are doing all this revenue generation out of that as well. So we want to move to very lean operation. We want to move to about a 3,000 to 4,000 square feet space and expand our manufacturing and business development teams. So and that will involve buying some equipment and hiring people and leases. So most of the proceeds will actually go towards direct revenue generation activities.
And what is the minimum investment amount that you would accept?
So minimum investment is about 250,000. That's what we are looking for right now. More the better. I want to keep the cap table simpler, but also, as I said, minimum. If there are investors who can add more value beyond a check size, then we are open to considering smaller too. But it has to be, other values along with it, other value along.
Right. And lastly, with all of your experience in biotech, what attracted you to this opportunity at Novel?
That's a good question. So during COVID pandemic, we were involved with Operation Warp Speed, and at that time, I had my other two business. They're still operating profitable entities, and I've kind of sold my stake. I'm a minority stakeholder now in those two entities, so I've exited partially. But I was involved with that, and I was building plasmid DNA facilities for I won't disclose the names, but two very large companies, um, and and I kind of realized that DNA is becoming a big bottleneck. We're 2 years of lead time, which was very painful. And that was also one of the direct contributors to the vaccine rollout being delayed. And also other therapies were getting delayed. So as an engineer, as a GMP, and as a biotech professional, I started looking why this is the case, and realized that people are using basically 50-year-old technology, and there is no differentiation. And the response of the market to this challenge is my facility is bigger than yours. So we were building bigger facilities, and bigger facility comes with a price tag, and that kind of makes it more complicated. So you're not actually solving the problem. So, I used to think in my mind that, when the car was invented, there was shorter distances, simpler roads, and we developed an engine. And now we have long highways, and we can go higher speed. We need a better engine, but we're still using an engine from the 70s. So we're just building a bigger and more shinier car with the same engine. So why don't we change the engine itself? Why nobody's looking at changing the engine? So that kind of thought process I pondered over for some time, and started reviewing some research into other bacteria that are available that can do this job, and pondered over a research paper written by my now CTO, Matthew Weinstock, where he had demonstrated the bacteria that has a lot of potential. And that kind of intrigued me, and I put my own money into it. And I wanted to solve this problem for myself and for others. And we did a proof of concept that demonstrated, yeah, this bacteria can really work. And then as we move forward, we got into the same problem that others would have, that nobody knows how to work with a new bacteria. So as we tried to mutate it, it resisted. So we kind of got stuck. So at that time, I approached Matthew Weinstock, whose paper I had read, and he was coming out of AppSci. He became the CTO of AppSci at that time, and he took the company public, and he was available. And he was very excited about this bacteria and the potential of this bacteria. So he joined us as a CTO, and he came with him with about 6 or 7 years of experience already working with this bacteria. And that kind of fast tracked our development journey. And we quickly kind of started developing this platform. So that is the overall kind of the summary of my experience. It actually started with not an idea in my head. It started with the real problem that I saw with my own hands and my eyes, and I was part of that and trying to solve that problem.
Great. Thank you so much. And thank you, everyone, for the great questions and for joining. Everyone out there has my email address. Please reach out to me if you want additional information, have additional questions. And in turn, Novel Bio will have all of your information as well, and we'll be reaching out. And I just want to thank everyone for joining, and thank you, Pankaj, for joining us today and giving us this great opportunity. And I hope everyone has a great day.